Well, hello and welcome to our first topic, which is cell biology. It is quite a big one. You'll notice it covers 3 weeks. And a lot of the things that we're going to talk about underpin a lot of the systems that we then talk about in more detail. So these are the basic processes that you need to get under your belt before we can start talking about specific systems and processes. It's split into 3 parts to cover the 3 weeks. This first part, we're going to look at the 2 major processes that synthesize key elements in terms of physiology. Firstly, we're going to look at proteins, synthesizing proteins. Proteins are ubiquitous. Channels, receptors, chemicals, they're all proteins. And then we're going to look at synthesizing our little batteries, our source of energy. We're going to look at ATP synthesis. So that's in this first part. Enjoy. So let's start with protein synthesis. In protein synthesis, there are 2 main phases occurring in different parts of the cell. In transcription, a copy is made of one of the strands of the DNA helix. In translation, that copy, the template known as messenger RNA, is then fed through ribosomes outside of the nucleus, and amino acids become bound together to form a simple polypeptide chain. This is the start of our simple protein. After that, the polypeptide chain can be processed in different ways, and it ends up becoming a functional protein. The protein can be used within the cell. It can be mounted on the plasma membrane of the cell, what we call expressed by the cell, or it can be released completely into the circulation. The protein can be a chemical. So we could be talking about a hormone or an enzyme or a neurotransmitter even. It could also, the more complex folded polypeptides with multiple polypeptide chains put together, bound together, it can become a structural protein. So we're talking about something like a receptor or a channel or various other structures around the cells or outside the cells. So let's go back to the beginning and talk about DNA. You did a little bit on this in your pre-reading. DNA, as we know, is a double helix. Each strand is a sequence of nucleotides, 4 nucleotides in case of DNA, on a sugar phosphate backbone. The nucleobase that contains a nitrogen group, hence it's called nitrogenous, determines the type of nucleotide. So, for example, adenine, but also in the list there you see thymine, cytosine, and guanine. A sequence of 3 nucleotides in a row forms a codon. Each codon codes for a specific amino acid. And the strands, the 2 strands of the helix, are arranged so that the specific nucleobases pair to each other. So there is a definitive way of pairing. Adenine pairs to thymine. Guanine pairs to cytosine. On RNA, thymine is replaced by uracil. So adenine there will pair to uracil. So it's A to T or U and C to G. Easy way to remember this, I always think, is A and T are both spiky letters, And C and G are big curly roundy letters. So A to T, C to G. Adenine to thymine or uracil, guanine to cytosine, and of course vice versa. Thymine pairs to adenine, cytosine pairs to guanine. So let's have a look at some diagrams. So here's our double strand of DNA. It's not twisted around in a helix because it's easier to look at when it's straight like that. And you can see, looking at that, we've marked out our 3 nucleotides that form our codons. So in this particular example, we have what? 1, 2, 3, 4, 5 codons. Also notice that the top strand is labelled the DNA sense strand. The bottom strand is labelled the DNA antisense strand. So it is this bottom one that we're going to pair the ribonucleotides to. And we're going to pair adenine to thymine, guanine to cytosine. Or in the case of where we're pairing to RNA, then adenine is going to pair to uracil, and uracil will pair to adenine. So, okay, RNA polymerase comes along, our transcription factor binds, that initiates the whole process. In comes this big enzyme, RNA polymerase. First thing it needs to do is unzip those strands, so it separates the 2 strands apart. We've now at the bottom got our DNA antisense strand exposed and ready to bind ribonucleotides. So RNA polymerase then recruits those ribonucleotides, and they come in and they pair with the antisense strand. And if you look at the corresponding nucleotides on the sense strand at the top there, you'll notice that this is a copy, other than the fact that our T, because this is ribonucleotides, is replaced by U. So that carries on along the whole exposed strand. Eventually, we get a whole strand of messenger RNA. And again, as you can see, it's a copy of the DNA sense strand because it was paired to the antisense strand. That messenger RNA is now our template. It moves out of the nucleus via the little nuclear pores. And generally, the other side of the nuclear pores, we have our endoplasmic reticulum. Closest to the nucleus, it's called rough endoplasmic reticulum because it's studded with ribosomes. So this is where we now go into translation. So we transcribed the DNA. We took a copy of it. Now we need to translate that copy to make a protein or a polypeptide chain in the first instance. So translation occurs in the ribosomes within the rough endoplasmic reticulum. And there you can see in the diagram, this is what leads out from those nuclear pores from the nucleus. Ultimately, the rough endoplasmic reticulum moves through little corridors into the smooth endoplasmic reticulum. So it leaves the ribosomes behind. Translation involves obviously our messenger RNA, otherwise it would be pointless making it. So as we said, this leaves the nucleus via the nuclear pores and enters the rough endoplasmic reticulum. It also involves another form of RNA. This is ribosomal RNA. Ribosomal RNA, or rRNA, forms most of the ribosome structure. And ribosomes have two subunits. They have a large subunit and a small subunit. And in the top picture there, the ribosomes are represented by the little orange dots. And there you see a close-up of the complex shape of the ribosome. We also have another form of RNA. This is transfer RNA, or tRNA. And this is the structure that carries the specific amino acids in from the cytosol to all line up to form our polypeptide chain. And it does that by pairing through our nucleotides again, our nucleobases. It pairs to messenger RNA. So transfer RNA has two distinct ends. At the top there, you can see what we've got is a binding site for a specific amino acid. In this case, I've put in alanine for the sake of argument. The amino acid that binds at that end is determined by the anticodon on the other end. So at the other end, we have an anticodon. So these are nucleobases again, but they are the ones that will pair to the codons on messenger RNA. So we have three little binding sites for specific codons. That sequence of nucleobases determines the amino acid that can bind at the other end of transfer RNA. So if this transfer RNA is specific for the amino acid alanine, as I've got at the top there, the anticodon could be GCU, GCC, GCA, or GCG. We'll see in a minute why there's multiple options for, in terms of codon, for amino acids. So each amino acid is coded for by different codons. So the anticodon pairs to the codons on messenger RNA in exactly the same way, A to U, C to G. So amino acids and codons then. roughly 20 amino acids make up human proteins. They string together and they form the basis of all the proteins. Two of the others are a little bit controversial, and some of them, they're only seen at certain times, some of them only when we're ill. You suddenly see a couple of other amino acids, but generally we have about 20. 9 of them are essential. This means that you can't make them. They have to come from food. The remainder, we can synthesize them. And amino acids are basically molecules, and those molecules contain carbon, oxygen, hydrogen, and nitrogen in different quantities. The R group you see at the top there, you see an R. There is no chemical that has R, as far as I'm aware, not one that makes up amino acids. That's the R group. The R group determines the specific amino acid. So whatever is bound on there will decide what sort of amino acid it is. Because of that combination, those pairing possibilities, there are 64 possible codons, including 3 stop codons. So we have 61 codons coding for 20 amino acids. The stop codons, incidentally, you don't have to remember this, are UAA, UAG, and UGA. Each of the other 61 codons codes for a single amino acid. But yes, there are more codons than amino acids. So this is why several codons may code for one amino acid. We look at that in a table. You don't have to remember any of this, but it's just to give you an idea. So if we look there at arginine, second from the top, then we can find its abbreviation on the chart and we can work out what the first base, second base, and third base, in other words, the first nuclear base, the second, and the third are. There you can see in dark red the stop codons. So if we really wanted to know what arginine was, and I'm trying to spot it here, you probably already have, there it is over there, we can see that we have various codons that can code for that. So we have AG and then possibly another A or a G. As I say, you don't have to remember all this. This is just to demonstrate that although we have a limited number of amino acids, we have more codons. So multiple codons can code for a single amino acid. So let's put all those components together and talk about the process of translation then. If you remember, our messenger RNA came out of our nuclear pores into the rough endoplasmic reticulum. It then binds to the small subunit of a ribosome. Remember, we have two subunits in ribosomes, small and large. The large subunit then binds, and that sort of holds the messenger RNA in place. It traps it between the two subunits. Messenger RNA is then fed through the gap between those subunits, so it's fed through the ribosome. As it's fed through, a sequence of transfer RNAs, remember transfer RNA has the anticodon one end, has a specific amino acid the other end. So as it's fed through, a sequence of transfer RNAs, tRNAs, pair their anticodons to the messenger RNA codons, one at a time. At the other end, as we said, is the specific amino acid for that anticodon. The amino acids, as they line up, get bound together via peptide bonds by the ribosome. The transfer RNA then leaves empty-handed, goes away leaving its amino acid behind, and it might go off and find another amino acid that it's specific for to bind to it. Or it might just go off and have a little rest. The bound amino acids then form the polypeptide chain. Eventually, once we get to the stop codon, that'll detach from the ribosome, and at that point the whole messenger RNA strand has been translated. So let's have a look at some pictures. Here's our small ribosomal subunit. So if you remember, messenger RNA is going to come along and bind to that. Here he comes. There's messenger RNA. Our large subunit now comes in and clamps that messenger RNA in place. Along comes transfer RNA, a slightly stylized transfer RNA, but you get the idea. At the top of it, you can see it's got its anticodon that's going to pair to the codon on the messenger RNA. The other end, it has an amino acid that is specific to that anticodon. The transfer RNA then goes away, leaving behind its amino acid. In comes another transfer RNA with another amino acid. In this case, we've got a different one because that's determined by the anticodon on this particular transfer RNA. And the same thing happens again. Off goes transfer RNA, and we form peptide bonds. The ribosome creates these peptide bonds to hold those amino acids together in what is going to be a polypeptide chain. So at the moment we have a peptide. We have two amino acids joined together. What we want is a great long chain. So that will continue until such time as we get our full chain. So once we get to the stop codon, messenger RNA has done its job, and what we'll have is a polypeptide chain of amino acids, all joined together by peptide bonds. And you can see there some of the amino acids may repeat. They could even be next to each other, the same amino acid. So protein processing. Well, so far all we've got is a simple polypeptide chain. And if you think particularly in terms of structural proteins, we need to do quite a lot of processing to turn that into something useful. So within the endoplasmic reticulum, so further along in the smooth endoplasmic reticulum, and then within vesicles that bud off from the sarcoplasmic reticulum, and then within a structure called the Golgi apparatus, the polypeptide chains are processed into proteins that may form structures or chemicals. And remember, all of these organelles can interact, and they can interact with the plasma membrane because all of those membranes within the cell are phospholipid bilayers. They're exactly the same. So they can all fuse together or move away from each other. The type of processing that might happen might be the joining of multiple polypeptide chains to make a more complex protein of multiple strands. It may involve the adding of sugars to make glycoproteins or lipids to make lipoproteins, or even in some cases both to get a glycolipoprotein. So we get all these things added and we get chains added together and bonded together. Another thing that might happen is some folding. So folding can be really simple. We can end up with just an S shape with a couple of bonds holding it together into that S shape. Or we can get some really complex structures. You remember we saw the ribosome just now. The ribosome, you could see it was multiple chains all folded together tightly in specific ways to make it function. There also might be some cleavage, and this is a classic activation of some chemicals that are chemical proteins that need to be activated before they can be used. And often activation involves in chopping bits off or halving them. So you get cleavage of sections from the protein to activate it. We'll look at an example in a minute. These proteins are then stored in vesicles for later use, or if they're secreted immediately after they're synthesized, then they will be exocytosed from the cell. And we'll talk about exocytosis in part 2. So a polypeptide chain there. You can see on the left we've got this simple polypeptide chain. We can then do some folding, and it can get folded into specific shapes. And there you can see something called a beta pleated sheet and an
alpha helix. You can then put those together, and this then takes us to our tertiary protein structure, where we have all that folding put together into a complex lump, if you like, of protein. And eventually, we might get more than one amino acid chain involved. We might get two of those complex folded structures put together or more. And we get our quaternary protein structure. So we've got levels of complexity going on there. We talked about cleaving, and a great example of this really is protein hormones. So here we've got something called pre-pro insulin. Yeah, it's the hormone insulin, but this is its early phase, pre-pro insulin. It's inactive at this stage. The next thing that happens is that little bit at the end, the signal sequence, is removed. It's cleaved, not with a knife like that, but you get the idea. That's cleaved off. Cleavage is usually done by enzymes, which of course are also chemical proteins. Once that's cleaved off, we get something called proinsulin. We're still not got active insulin. So at a later stage, we get this midpoint, proinsulin. We then cut that completely in half. When we cut it in half, we end up with this C-peptide chain, but we also end up with active insulin. So all protein hormones really are processed in this way. They start as pre-pro hormones. They become pro-hormones when the signal sequence is cleaved off, and then they become active hormones right at the end. So that's cleavage. So that was our protein synthesis and processing. So that made a massive component of the body. Obviously lots of little components, but a large part of us is made up of proteins. Another thing that we use in large quantities is energy. So cellular respiration is about making ATP. ATP are like our little batteries, and those little batteries can be recharged, and we'll talk about that, if you like, recharging process. So, as we said, cellular respiration produces ATP, adenosine triphosphate. You can abbreviate it throughout this module and subsequent modules to ATP. There's no need to write that out in full. It's such a well-known abbreviation. And as we said, it's the energy unit used by many cells for thousands of processes. So the adenosine part is, there you see the blue shapes there, is adenine. Adenine is a nucleobase, if you remember. Adenine pairs to, thymine or uracil. So we have adenine, and bound to that we have a 5-sided pentamer ribose sugar. Those two bound together give us adenosine, and adenosine can exist in that form. We will come across it later in clinical reasoning in particular, and its role there. Adenosine triphosphates, though, is where we take adenosine and then by high energy bonds we attach to that 3 phosphate groups. So we have phosphate and oxygen forming these little phosphate groups. 3 of those bound together by high energy bonds gives us a TP, triphosphate. phosphate. In between that, we can have adenosine monophosphate, AMP, and adenosine diphosphate, ADP. And generally, when we use the energy from ATP, what we end up with, apart from energy, is a DP and a free phosphate. And the energy is all in breaking those bonds. So the bonds between adenosine and the phosphates and between the phosphates themselves produce energy when they're broken by hydrolysis. So it's nothing to do with the phosphates themselves. They're just something that is bound through this high energy bond. It's the breaking of the bond that gives the energy. We also have, and we will come across it in part 2, guanosine triphosphate. Now, ATP is generally our source of energy. There is only one process that I can think of that uses GTP for energy. It's exactly the same, but here we have guanine rather than adenine. So the energy from hydrolyzing these bonds between the phosphates, and here we're usually only talking about that first bond between a DP and that final phosphate, is used by cells for many different processes. So we hydrolyze this bond and it releases energy. We then left with ADP and a phosphate that just hangs around inside the cell. Cells are full of phosphates. So the ADP then has to be bound to another free phosphate during cellular respiration to synthesize more ATP. We could break the next bond between AMP and that second phosphate, but that wouldn't release as much energy. And the bond between adenosine and the first phosphate, even less energy. So ideally, we don't want to do that. And we don't usually have to do that. The whole system is so efficient that really we only generally hydrolyze that first high energy bond. So we end up with ADP, and we need to put another phosphate back on it to make more ATP to replace the one we just used. Each ATP is used and rephosphorylated within approximately 1 minute. You'll find throughout this module there's loads of things that take me quite a while to explain or draw or whatever, and they happen in milliseconds in many cases. So within 1 minute you can do that, hydrolyze that bond, and then stick another phosphate on, and it's ready to go again within a minute. Best battery charger I know. We actually use 160 kilograms of ADP. Think how small it is. I don't know what it weighs, but it will be very light. 160 kilograms of ADP is used and phosphorylated every day. Obviously, that's going to change if we do more energetic things, but that's an average. So that's a lot of ADP to phosphorylate. In cellular respiration, there are 3 main stages, and you will have heard of at least the first 2, I should imagine. The first one is glycolysis. Glycolysis is anaerobic. In other words, it doesn't need oxygen, and it occurs in the cytosol of the cell. It also doesn't produce much ATP. The Krebs cycle, or citric acid cycle, occurs in the matrix in the middle of the mitochondrion. And again, it still doesn't produce much ATP. The final phase, the one that produces about 90% of the ATP, is the electron transport chain. This occurs across the inner membrane of the mitochondrion. So a mitochondrion has two membranes and an intermembrane space. That inner membrane is where the electron transport chain occurs. And it produces most of the ATP. Really, glycolysis and the Krebs cycle are all about providing the requirements for the electron transport chain. Every cell, apart from erythrocytes, has hundreds or thousands of mitochondria. And if you remember, mitochondria are able to replicate. So if you are expending a lot of energy over a period of time, you will make more mitochondria to supply those increased energy needs. So let's start with glycolysis. As we said, glycolysis occurs in the cytosol of the cell, and it's different to the other two phases in that it's anaerobic. It doesn't require oxygen. In glycolysis, we take glucose, which is a sugar with 6 carbons. It's a large molecule. It's broken down into 2 molecules of pyruvate. Each of those have 3 carbons, because everything has to go somewhere. So we produce pyruvate, 2 molecules per molecule of glucose. So there you can see our equation for glucose, and you can see where everything is going. We have a couple of H left over, so we have a bit of excess of that. But that's the chemical equation of pyruvate C3, so 2 of those taking 3 of the carbons each. H4, so we've used 8 of those 12 hydrogens, and each of them have 3 oxygens, using up all that oxygen within the glucose molecule. This uses 2 ATP, so the conversion of glucose to pyruvate uses 2 ATP, but it produces 4. So we gain 2 ATP, which is not a lot in the grand scheme of things. More importantly, a coenzyme called NAD+, the abbreviation is absolutely fine, also gains electrons because it is an electron carrier. That's its role. It gains electrons. When it gains electrons, it becomes NADH. Again, it's an acceptable abbreviation. I'm not asking you to remember the full name of that. So NAD plus H plus 2 electrons. So if you notice there, in NAD we've got 1 plus, in the H plus we've got 1 plus, so we're able to carry 2 electrons to make that neutral. NADH being neutral, it has neither a plus nor a minus. And that reaction is reversible. It has to be reversible because this is an electron carrier. Just like a carrier bag, it'd be no point if you put your groceries in a carrier bag and then could never get them out again. So it's a reversible reaction. So created during glycolysis then, 2 pyruvate, we're going to do something with those in a minute, 2 NADH, we will do something with those in the electron transport chain. 2 ATP, yeah, we could use that. 2H plus, also useful in the electron transport chain. 2 molecules of water and some heat. So that's what we create. Pyruvate, NADH and H+, as I said, are necessary for the next phases of cellular respiration. And that all then moves into the matrices, the matrix of a mitochondrion, or matrices of the mitochondria. And that's really all you need to know about glycolysis. We're not going to go through it in any detail other than that, because at the end of the day, it doesn't produce much ATP anyway. Okay, so yeah, I fibbed a bit. There is another little phase in between glycolysis and the Krebs cycle. This is the intermediate phase, where we have to convert that pyruvate into something else first. We have already moved into the mitochondrial matrix, so we're in that pinky area in the middle of the mitochondrion. And what happens is pyruvate is converted to acetyl-CoA. Also releases carbon dioxide, so therefore we know it must use oxygen. It's an aerobic process because everything inside the mitochondrion is aerobic now. During this, a further NADH is produced. So we take NAD+, add some electrons, some hydrogen, and we make NADH. And as I said, eventually we're going to use all this NADH for the electron transport chain. So we've got acetyl-CoA. Carbon dioxide that's produced is excreted from the cell by diffusion, simple diffusion, which we'll talk about in part 2. When you have a situation where you don't have much oxygen, you're hypoxic, pyruvate, of course, we're now using oxygen. So you can't convert pyruvate to acetyl-CoA without oxygen. So what actually happens in the simplest of ways is pyruvate is fermented in the cytosol. Again, we're not going into details, to produce lactic acid. NADH, because we now don't need it for the electron transport chain because we're going to need some oxygen for that as well, is reconverted back to NAD+. And of course, this is going to severely limit the amount of ATP produced. And often in hypoxia, it's when we will start to hydrolyze more of those bonds. So we might get down to AMP rather than ADP. So our 2 acetyl-CoA, we made 1 per pyruvate. Remember, 2 pyruvates per glucose molecule gives us 2 acetyl-CoA. Then enter the Krebs cycle. And again, we're not going to go into too much detail here. The Krebs citric acid cycle is also aerobic. It's still within the mitochondrial matrix. Each of the 2 acetyl-CoA molecules goes through 8 enzymatic reactions. And you will see in your textbooks lots of complex circular drawings with arrows and lots of enzymes and all the rest of it. For this module, you don't need to know that. It's really not that important. What is important is what it produces. So again, it produces key solutes for the electron transport chain. So each acetyl-CoA produces 3 NADH, so our electron carriers. It also produces 1 FADH2. This is another electron carrier. Different type. Same job, different type. And 3H+. Those are the things that we want for our electron transport chain. We also get one ATP and one GTP, guanosine triphosphate. We get carbon dioxide. Carbon dioxide again diffuses out of the mitochondria and out of the cell as waste. Which brings us to the electron transport chain. So theoretically, and you will see different figures in different textbooks, 36 ATP molecules can be made per oxidized glucose molecule during cellular respiration. But that's not usually the case. There's often losses due to leaky membranes or the cost of moving pyruvate and ADP into the matrix. So the average is around 29 to 30 ATP per glucose. That's a lot of ATP per Mars bar. But actually, because this is so efficient, 50% of the energy produced is usually released as heat energy. The electron transport chain, though, provides 90% of the ATP. It uses H+ to activate an enzyme called ATP synthase. The use of H+ to do this is called chemiosmosis. Not osmosis as in water, chemiosmosis. The whole of the electron transport chain involves oxidation, and it results in the phosphorylation of ADP. In other words, the adding of a phosphate to ADP. Hence, the electron transport chain may be called oxidative phosphorylation. And you may see this again in textbooks. So here's a diagram. So if you look at the bottom, you'll see I've got the word matrix. That's the matrix of the mitochondrion. Above the phospholipid bilayer, I've got the intermembrane space. And right at the top, we've got the outer membrane of the mitochondrion. So we've got matrix at the bottom, inner membrane, intermembrane space, outer membrane. And you'll see I've got some green blobs and I've got a purple blob. And we'll talk about what those blobs do in a minute. Okay, the green blobs are proton pumps. Now, the word proton here, we're talking about H+. H+ has one proton in its nucleus and no electrons, because H, hydrogen, as an element, only has one electron. If it loses that negative electron, it becomes positive. It also has no electrons and one proton. Hence, people often refer to H+ as protons, not to be confused with the subatomic particle within the nucleus of atoms. This is proton used to refer to H+. So hence we have proton pumps. Those proton pumps are going to transport H+ from the matrix of the mitochondria into the intermembrane space. Between them, the darker green small blobs, we've got carrier proteins. These carrier proteins basically perform the simple function of enabling the electrons that we're going to put in here in a minute to move from one proton pump to another along the chain. They're really just there to sort of facilitate the electrons' movement. Our purple blob, this is where H plus is going to return from the intermembrane space back into the matrix of the mitochondrion. When it does that, it turns the rotor on the top there, and this then activates the ATP synthase below it. Synthase, of course, means it's an enzyme because it ends in ase. Synth, clues in the name, it synthesizes ATP. ATP synthase. The rotor activates ATP synthase. ATP synthase then adds a phosphate to ADP. It phosphorylates ADP, which of course is going to give us ATP. So let's look at that as a process. First of all, you need to remember that proton in this case, as I said, refers to the cation H+. It is an atom of hydrogen that has lost its only electron. Therefore, it's got a single proton, and therefore people call it a proton. Electrons, on the other hand, are negatively charged subatomic particles, where we're talking about them here. So usually they circle the nucleus of atoms, but they can be released, and they're usually released in redox reactions, reduction oxidation reactions. You don't really need to know about those. If an atom loses an electron, it becomes more positive because it's lost negative charge. Then it's called a cation. So an example might be H+, Na+, K+. If an atom loses 2 electrons, it can become a double cation. And key examples in the body are certainly calcium, Ca++, we will come back to, or Fe++, iron. And this is the way I want you to write these cations. If you write the word calcium, then it means the atom, it doesn't mean the ion. You could write calcium cation, but that would take more writing, and also it would indicate Ca+, not plus plus. You'd have to write calcium double cation. It's much easier to write Ca++, or Fe++ for iron, or H+ for our hydrogen cation, Na+, sodium cation. Cl- is a chlorine anion. It has gained an electron, so it's become more negative. But we don't write the full words, we write the symbols. Okay, so back to our process. So here's our proton pumps, our carrier proteins, our rotor on top of ATP synthase. So here comes NADH. Remember, this is our electron carrier, or one of our electron carriers. It comes into that first proton pump in the chain. It drops off its electrons. Remember, we said that equation was reversible. This is why. We now need to reverse it to release the electrons that were being carried. So it drops off the electrons at the first proton pump. NAD+, of course, is what's left over. That can go away and can go and get some more electrons. That's done its job. those electrons now start to move through the chain. So they move through the proton pumps, assisted by the carrier proteins between the proton pumps. What they're doing is they're driving those proton pumps. They're making them work. They're activating them. FADH2, remember, was our other. We have fewer of them, but it's another electron carrier. So FADH2, don't forget the little 2, it's got 2 hydrogens, drops off its electrons at that first carrier protein, or more or less round about there. And that leaves FAD. FAD, again, goes off, can gather up some more electrons, do its job again. Everything gets recycled. And they keep on moving through. So they move through the chain of pumps, these proton pumps, assisted by the carrier proteins. Eventually, we need to mop up those electrons because you can't leave electrons floating around. They're a bit dangerous. They react with things. So what we do is we mop them up to create water. So 2H plus mop up the electrons by combining with an atom of oxygen. Remember, O2 is a molecule. It's 2 atoms of oxygen bound together. So here we're talking about an atom because we've only got one O there. We've got 2 H's, 1 O, H2O. Those H's were H pluses, but because we've chucked in a couple of electrons, they're now neutral. So we have H2O. This is not really part of the process, it's not producing ATP or anything else, but it's a way of mopping up those electrons. But what are our electrons doing with those proton pumps? Well, they're pumping protons. The proton pumps are driven by the electrons or activated by the electrons. And what they do is they pump H plus from the matrix into the intermembrane space. So they pump one way. Generally, pumps are a form of active transport, which we'll talk about in part 2. And generally, active transport involves the use of ATP. But in this case, that will be a bit counterproductive because, of course, we're trying to make ATP. We don't really want to use it to operate the proton pumps. So this is where the electrons come in. They're providing those proton pumps with the impetus to pump the H plus, the protons, from the matrix into the intermembrane space. Eventually, H plus accumulates in the intermembrane space. At the moment, it's got no way of getting out. It's stuck there between that inner and outer membrane of the mitochondrion. It's trapped. And what happens is this creates an electrochemical gradient. Because there's more H plus, we'll talk about these gradients a bit later, but because there's more H plus in that intermembrane space than there is in the matrix, it wants to equalize. It wants to go back to the matrix. It wants to follow a gradient back into the matrix. And that gradient is both electrical, because of the charge, and chemical, because we've now got more hydrogen, the chemical, inside that intermembrane space than we do in the matrix. So it wants to return to the matrix. So it does, because it's sitting there and it suddenly finds, oh, hang on a minute, there's a little hole, there's a pore through this little rotor thing I could go back through that, and it does. It follows its electrochemical gradient back through that rotor. As it goes through the rotor, it spins it. Really funky little mechanism. So as it's passing through, it's causing that rotor to spin. And the spinning of that rotor is what activates the ATP synthase. So ATP synthase is activated by the movement of H+ back through the rotor, which spins the rotor, which activates ATP synthase. ATP synthase does its job. It synthesizes ATP. So in other words, it takes ADP, adenosine diphosphate, 2 phosphates, and a spare phosphate. Remember I said there's loads of them floating around. And it combines those together to make ATP. So it phosphorylates ADP. It adds a phosphate to ADP, which is going to give us ATP.
So well done, you've completed part 1 of cell biology. That was all about the synthesis of proteins, the synthesis of ATP. Before you move on to part 2, access the full PDF version, and if you want to, you can print it, you can download it, read it digitally, whatever you want to do. But there is a written version, and that's all 3 parts in one document. Access any of the suggested animations or other resources related to this part. Also, remember to join our live session as you've been allocated, and we can go over it with a whiteboard in a bit more detail, discuss it, answer your questions. But also start to make your own notes now. A glossary is a great thing to have. You will find there will be lots of terminology coming up throughout the module. And having a glossary where on one side you put the word, on the other side you put a definition, it gives you the ability to cover one side and try and remember what the word is or what the definition is. So having a glossary is a great thing to have. Writing your own notes in your own words, bullet points, the sort of notes that are portable that you can take with you, so you're standing waiting for the bus for 10 minutes, you can have a look through and start to learn the material, because it's not just about understanding this, it's also about learning it. Most students say, I have no problem understanding it, it's the remembering it. And that's where I can't do the work for you. You have to put the work into that. And it's known that if you use multiple pathways to do that, so reading, writing, speaking, drawing, then you will form those memories better for actual physiological reasons. So do all of those things. Write your own notes, do your own drawings. Start to think about speaking it, if only to yourself at first, or the cat, or your husband, or your children, or ideally colleagues at work who are doing the same course or have done the course. Try to teach them the concepts that you're learning. By doing that, it will help you to remember it. So start to get those techniques done and make sure you put the hours in to learning this. Otherwise you're going to get left behind very, very quickly. And now for part 2.